So we're honoured to have Krishnendu, visiting fellow at Dr. Tom Mistelli's lab at National Cancer Institute, National Institute of Health, Bethesda, Maryland, who's going to share his insights into the role of centromeres in 3D genome organisation across species in Canada. Thank you very much, Krishnendu.
Thank you, Caroline. So let me find my slide, share the screen. So can you see my screen?
No.
Okay. What about now?
Yes.
Okay.
Yeah, now we can. We can see your screen and the slides on the left hand. There we go. Now we can see presenter view.
Awesome. So thank you for the kind introduction, Kathleen. I hope I am audible and you guys can hear me properly. So today I'm going to talk to you about one of the projects that I pursued during my PhD. And this is a story about the fungal genome evolution that happened about like 39 million years ago. So this is a story I'm going to tell you, and I hope you like it. So let me start by defining what centromeres are. And the centromeres are the genomic loci that specifically binds to the kinetochore proteins. And these kinetochore proteins, in turn, attaches to the microtubule fibers from opposite poles during the mitotic cell division. And they segregate the replicated genome into two different daughter cells. So this process of centromere-mediated genome segregation is kind of conserved across different species, if you look at it. But the interesting point is that the centromere DNA sequences, the underlying DNA sequence elements, they are not conserved across species, although this process is conserved. So what I'm showing you here is a figure where you can see that the centromere DNA sequence of different species can be highly variable in terms of their DNA sequence length, the organization of the repeats, presence of the repeat structure and organization both can be highly diverse across different species. And this is known as centromere paradox in the field, why the centromeres are not conserved, although the process of centromere mediated genome segregation is conserved. So this idea of centromere paradox, I first got exposed to this idea during my PhD in Kostov's lab. And I could finally work into a project that kind of related to this idea of centromere paradox, and I'm really glad that I could work on that area. So when I looked at the fungal centromeres, the centromere sequences across the known and sequenced fungal species, there are about 50 fungal species that are now studied for their centromeric sequences and properties. So the interesting point is that the centromeres in fungal species are not conserved in terms of their length and as well as the repeat content, organization of the repeats. They're very diverse, similar to the other species that I was talking to you about earlier. So this diverged properties of centromere sequences is something that I kind of find that this idea of centromere divergence is kind of interesting in terms of evolutionary perspective. And when I joined Kostov's lab, my senior Gautam was working on identification of centromeres in Candida tropicalis, which is, of course, a potential threat and emerging drug-resistant species in Indian subcontinents. So he was working to identify the centromere of Candida tropicalis, and he did chip sequencing for the kinetochore proteins. And he showed that the centromeres of Candida tropicalis are 7 let me try switching to the laser pointer. So if you see the structure of the centromeres in Candida tropicalis, they're all inverted repeat associated structures. And the interesting point is that all 7 centromeres have high sequence identity, as you can see here, between 80 to 90% sequence identity between the 7 centromeres in the species. So the interesting thing is, although there are 7 centromeres, and this is kind of conserved, this is not only true for Candida tropicalis, but also other species in Candida subcomplex and also in Saccharomycotina, that the centromeres always clustered at single point, as you can see here. This figure shows the centromere localization of different kinetochore proteins, NPA, NPC, NAF2, and DAD1. And if you look at the interface cell or cells that are going through mitosis, you will see that there are all the 7 centromeres, they're clustered together in one spot. And when they segregate, you see 2 spots per cell. So even though they have 7 centromeres, they're all clustered at one point, like I have shown here in this figure, that you can see all the centromeres are clustered. And this is also known in other fungal species that we reviewed earlier. So this identification of this inverted repeat associated structures in Candida tropicalis was kind of surprising for us, because before identification of centromere in Candida tropicalis, the other two species with known centromere, the centromeres of Candida albicans and Candida dubliniensis, they're kind of unique DNA sequences and without structural elements to them. Although one of the centromere in Candida albicans has inverted repeat associated elements. So all the centromeres of Candida tropicalis that has inverted repeat associated homogenized sequences was kind of interesting for us. And we asked that, these two group of species, Candida albicans and dubliniensis, and we have Candida tropicalis. So how did the centromere diverge so fast, and in terms of their sequence and as well as the structural elements? So, and what triggered this transition in centromere structures? So one of the clue to this hypothesis that I'm going to show you now came from the preliminary analysis between the genomic sequences of Candida albicans and Candida tropicalis. And that analysis showed that the centromeres are located as at the synteny breakpoints between these two species. So that kind of hinted us that the centromere might have gone through rearrangements in the last common ancestor, but since the genome assembly of Candida tropicalis was incomplete, we could not really claim that point earlier. And so we thought of this hypothesis at this point to explain how these centromere structures and inverted repeats were lost in one of the lineages, which is Candida albicans. And we kind of hypothesized that the last common ancestor had clustered centromere, which were also homogenized inverted repeat associated sequence. And if you have them clustered together, there are quite high probability that that break in one of the centromere leads to the translocation between the homogenized sequences of other chromosomes that are in physical proximity. So this is the hypothesis that we thought of at this moment. But to test this hypothesis, we needed to assemble the genome of Candida tropicalis, because at that time we did not have the genome assembly of Candida tropicalis in complete chromosome level. So and also Candida tropicalis, as I told you, it's an emerging species that is on kind of emerging as a multiple drug resistance isolates in clinical. Of course, you guys know better than me. So this is the idea that if you have genomic rearrangements in the chromosome level, genomic rearrangements in the species, fungal species, that often lead to drug resistance in this fungal species. So if we have a genome level, chromosome level genome assembly of the species, one can study the large karyotypic changes that are associated with the drug resistance in the species. So these are the two main motivations that led us to work on the genome assembly of Candida tropicalis. And we used 3C sequencing in collaboration with Dr. Amartya Sanyal's lab in Singapore NTU. And we did Pacific Bioscience sequencing, long read sequencing in association with, we got help from Joe Heitman's lab in Duke. And using these two data type, 3C sequencing and the Pacbio sequencing data, we could assemble the contig level assembly that was already available at the time, 23 contigs. We use these two data to build telomere to telomere long gapless 7 chromosomes. So this is the ideogram of the genome that you can see here. And they have like 7 chromosomes, right? So 1, 2, 3, 4, based on their length, they're named, I mean, they're numbered based on their length. So the largest one is chromosome 1. And as you go across the length, the numbers are so and so. And chromosome R is the chromosome which has the rDNA locus in the genome. So Candida tropicalis has only one rDNA locus in this chromosome. And so this is the final version of the assembly that I have showed you, and that was developed using these two data type. And then we thought we have to validate this by running Spatial. So here is the cleanest gel I could run in my PhD, and even then you could see only 4 bands here on the top: 1, 2, 3, 4, 5, 6. So where is the chromosome 7? I mean, there are 7 chromosomes, right? So this was kind of interesting, and we could do some southern hybridization in combination with the Spatial blots to show that there are structural rearrangements within the chromosomes, so that the 2 different homologs actually become of same size. So based on this data, we could validate our genome assembly that we developed. And using this genome assembly, we thought of first testing the 3D organization of Candida tropicalis genome, because we already did the 3C sequencing. So now we realigned the 3C sequencing reads on this chromosome level genome assembly of Candida tropicalis. And this is the matrix, the contact probability matrix that you see here. The color scale tells you that how frequent this contact can be in the genome. If it is white-ish, yellowish, then they are not probably interacting with each other. If it is towards the red, that means they're interacting with each other. So if you look at this matrix, you have the chromosome aligned on both axes of the matrix: chromosome 1, 2, 3, 4, 5, 6, and R. And the first thing we notice is that we have a strong diagonal in the matrix, which means that the sequences which are next to each other, the neighboring DNA sequences, they have the highest amount of contacts between themselves. And this is expected because they are just next to each other. What is not expected, though, is the interaction between centromere, at least from the perspective that the centromeres are present in different chromosomes at that, and the centromeres, they're not linearly close in the genome. So this interaction is happening in space. All the centromeres, they're spatially proximal, although they're from different chromosomes. So these spots that you see here, the centromere of chromosome 2, the spots means that the centromere of chromosome 2 interacts with the centromere of chromosome 1. And if you look at different chromosomes, you would see that the centromere of, for example, this centromere of chromosome 2 interacts with all the other centromeres in this genome, right? So this is what we could see from this analysis, and we could also see that there are some amount of interaction between the telomeres. As you can see here, I'm trying to show you with this highlighter, but it might be difficult to see. So, and it was difficult for us as well. So we did a aggregate sequence, aggregate signal analysis for centromeres and telomeres. So what we did is we pulled all the contact information from all the chromosomes, and we could see whether there is a trend in the genome. So this means that all the centromeres have higher interaction between themselves, and also you have enriched contact frequency near the telomeres. So this data tells you that the genome adapts kind of a rabble-like conformation in which the centromeres are kind of proximal to each other, like shown in this figure. And the telomeres are kind of away from the centromere. As you can see here, the centromeres do not interact with the rest of the genome. So if you walk away from the centromere to other genomic loci contact drops. So you see this cross-like pattern in the centromere. So this means the centromere is not interacting with other parts of the genome. And this rebel-like conformation kind of, we validated this with the imaging of Candida tropicalis nuclei stained with CNPA. These red spots are CNPA, and the genome is stained with DAPI. So you see that the centromeres are generally associated with the nuclear periphery, and they don't have contact with the rest of the genome. So with this observation of 3D genome organization in Candida tropicalis, we went back to the model and said that now we have the data for two different species. Candida albicans was known earlier, which has the unique centromere, and the Candida tropicalis, we did this 3C sequencing, and we showed that the centromeres interact in space. So now these two different species have the clustered centromere, which posits that the last common ancestor would have the clustered centromeres. And if they're clustered together, and that proximity guided translocation between the centromere led to the formation of the new centromere in Candida albicans, then this rearrangement should only happen near the centromere and not anywhere else in the genome. So now that we have the chromosome level assembly, we could test this idea whether the breakpoints are only at the centromere or they are also there in the genome. So we did the comparative genomic analysis between two different species, and you can see here the chromosomes of Candida albicans are color-coded. And if you map the specific ORFs from Candida albicans on Candida tropicalis genome, you would see that the centromere, which are highlighted with this triangle, little triangles here, all the centromeres of Candida tropicalis, they mostly present near the inter-chromosomal synteny breakpoints. For example, here you see this centromere of chromosome 2 is right at the synteny breakpoints, and the ORFs that are proximal to the centromeres in Candida tropicalis now, they come from two different chromosomes of Candida albicans. So with this observation that the centromeres in Candida tropicalis are associated with the inter-chromosomal synteny breakpoints, we could support the hypothesis that we presented earlier. And this observation also tells us that the last common ancestor went through this rearrangement specifically at the centromere, but why centromeres? Why not any other genomic locus? So we argued that the centromeres in last common ancestor could be the DNA sequence of the all 7 centromeres could be highly identical, and the sequence identity of the homogenized centromeres could lead to the could facilitate the translocations between the inter-centromeric translocation. So as you can see here, we have different species, the phylogeny of different species, and Candida tropicalis is here. You have Candida albicans here. So we have this last common ancestor of Candida tropicalis and Candida albicans here. So we are trying to show that this species, whether it has inverted repeat associated centromeres in the last common ancestor or not at this point. And to prove this point, we looked at the centromere DNA sequences for other Candida species, and we sequenced this species Candida soje and used the publicly available DNA sequences from NCBI database for Candida viswanati and Candida parapsilosis. And then, and then we scan through the genome to look for the DNA elements that matches with the signature of inverted repeat associated structure that we see at the centromeres of Candida tropicalis. So we look for the synteny structure, and the regions would be ORF-free or intergenic. Based on 3 criteria, we could predict the centromeres in Candida parapsilosis, and we could also predict the centromere in Candida soje, and we could show that the centromeres are inverted repeat associated structures. So this identification of putative centromeres that are inverted repeat associated structure in this Candida parapsilosis, which diverged earlier than this subcomplex, kind of supports the idea that this common ancestor between Candida tropicalis and albicans, they also had inverted repeat associated structure. So this identification of this inverted repeat associated structure in the last common ancestor, although we kind of predicted with the hypothesis, this is kind of unexpected because previously we did not really think that the centromeres of the subcomplex would have the inverted repeat associated centromere, because the centromere of Candida albicans and dubliniensis were kind of unique centromere DNA sequences. So when we thought that this last common ancestor might have the inverted repeat associated structure, we were interested to know how this DNA sequence of the last common ancestor could be. Is there any conserved DNA elements that you can predict from this analysis of known present day isolates of, present day species that we are having here? So we analyzed the DNA sequence of predicted centromeres from Candida parapsilosis, Viswanati, tropicalis, and Soji. And I should also mention here that when we were doing this work, the work from Geraldine Butler's lab could show that the centromeres from Candida parapsilosis are indeed the loci that we also found out by this sequence analysis. And they did the deep sequencing for kinetical proteins. So that kind of validates the observation that we had. So now we did the sequence analysis of these species that are having the inverted repeat associated structures. And this is a very crude way to represent the sequence, but as you can see here that the synteny dot plot, not the synteny dot plot, so the sequence identity between the species, you'll see that the dot plot analysis shows that these three species, Candida sojae, Candida tropicalis, and Candida viswanati, if you have this black spot on this matrix, that means they're highly identical. And this pattern, of course, means that they're inverted repeat associated. So you have this crisscross pattern that tells you that 7 centromeres in Candida sojae are also inverted repeat associated structure that are homogenized within themselves. And so observation of this highly homogenized centromere in this species complex, we also compared their individual repeat elements, the left repeat, the middle or the central core, and the right repeat, how similar they are within the species and across the species. And we could show that, compared to the random sequences, if you pick up 7 random sequences in the genome, they would probably have somewhere between 30 to 40% sequence identity, whereas the centromeres are quite conserved with more than 50% sequence identity, for example, between tropicalis and Candida viswanati. And that's also true for other combinations of Candida viswanati to sojae and tropicalis to sojae. So this species complex has some kind of conserved DNA elements, and we could show that there are some conserved DNA motifs within these sequences, although we did not really pursue that. And so what I have told you so far is that the sequences of the last common ancestor were kind of inverted repeat associated, and probably they were inverted repeat associated as well as homogenized within the centromeres. And that probably facilitated the rearrangements between the centromeres that led to the loss of homogenized repeats. So based on these arguments and analysis, we could show that the centromeres in Candida albicans emerged from the last common ancestor that had inverted repeat associated sequences. And when this last common ancestor went through inter-centromeric translocation, that led to loss of the homogenized inverted repeat associated centromere, and the unique centromeres were formed in Candida albicans that were kind of evolutionarily new. So this is the model that we proposed here, that the chromosome arms were extended at the centromere, mediated by the spatial proximity and sequence identity at the last common ancestor. So this is the story that I worked on during my PhD, and this was published in 2020. And when I finished my PhD, I was thinking about, like, if the centromeres are proximal to each other in fungal species, and that's true for most of the fungi, at least the one I have worked with, they are closely proximal, at least at some point of the cell cycle. And I was thinking, what's happening with the other species that, if the centromeres are proximal to each other and they go through this rearrangement, this can be beneficial for a species that, don't have the means of asexual reproduction, means of sexual reproduction and gender diversity within the population. But this kind of rearrangement gives them the way to evolve their genome and, fit into the landscape as a pathogen, right? But this is detrimental for the individual itself. You can imagine that the rearrangement when it happened, this is probably one of the lucky guy that survived and created all these new centromeres. But there must be ones that could not make this new centromere, and that would have been detrimental for them. They would have died. So if this happens in other species, in metazoans specifically, that would have severe consequences. And so I was interested in human genome organization because the human centromeres are not really known whether they're clustered or not. So now I'm kind of studying this organization of centromeres in human genome using different cell lines. Yeah, so I hope I could tell you some more stories about the human centromeres if I find one. I would stop here and thank Costa for all the support that he provided during my PhD, and my lab mates, Dr. Amartya Sanyal's lab from INTU Singapore, and Yao, who did the analysis for 3C sequencing data, and Bhagya from our lab. She also initially helped analyzing the DNA sequences. And also we got some help from Geraldine's lab. So thank you, everyone.
Thank you for that fascinating overview of what I know was an enormous body of work. I know that there'll be other questions from the group. I'm curious about you touched on the relevance to antifungal drug resistance, and we know that Candida tropicalis, as you've said, is much more likely to become azole resistant than Candida albicans. And I wonder whether it's related to centromeres. I know that one of the main mechanisms that Candida tropicalis will become resistant is through modification of its TAC1 ERG11 genes, which are on chromosome 5 near the centromere. So they can increase the copy numbers, or they can, have point mutations. Is that plausible? Is the difference in the centromere for Candida tropicalis more likely to lead to chromosomal structural changes?
When I joined the lab, I did not, of course, understand the conversation that went between Gautam and Kaustav, but we were all excited because, the centromeres of Candida tropicalis, all 7 centromeres have inverted repeat associated structure. And if you know about the story that came out from Judith Berman's lab about the isochromosome formation in Candida albicans that gives rise to fluconazole resistance. So the isochromosome formation happens on chromosome 5 of Candida albicans that has inverted repeat associated structure. So potentially, also theoretically, all the 7 chromosomes of Candida tropicalis can form isochromosome, right? So, yeah, but I don't know. One has to work and find out.
I think it was you, Julie, that asked a question along those lines when I early on when I was looking at the genome sequence. Sharon, you've got a question? You're on mute still.
Sorry, did Julie have a question? I'm very happy to.
Julie's
I didn't have a question.
Oh, sorry, Julie.
Go, Sharon.
Yeah. Thanks very much, Krishnendu. That was fascinating, and I won't pretend that I can understand everything. But in terms of people sequencing diploid species like tropicalis, what's your advice, and what are your tips for how to interpret the sequences, how to interpret, SNP variation, bearing in mind what we know about the 7 chromosomes and the centromeres, and how would you distill what is a true SNP from a false SNP?
Obviously, I did not talk to you about all the details of what we did for the genome assembly part, because Because of the time limitation, I did not want to bore you. But now that you talk about, we spend considerable amount of time to investigate the variation in the genome between the homologs and the SNPs, indels, also the large scale changes like the tandem repeats that we could find in the genome of Candida tropicalis type strain MYA3404. The one thing that I should say that in my experience, my little experience with this work is that you need a very good data set to work with before you can conclude anything. And also, if you are specifically pursuing the difference between SNP, I mean the real SNP and false SNP, I would say that you need at least another method of validation. For example, maybe Sanger sequencing, or maybe PCR, or maybe restriction digestion mediated confirmation, if you specifically chase specific SNPs. But I understand if you go genome-wide, then you need a lot of data and a lot of support in terms of sequence coverage before you can confirm that.
Okay, thank you.
Yeah, so more depth required
Yeah,
for a diploid.
yeah. But I also think that the sequencing technologies are constantly being improved by at least both at the chemistry and analysis of the sequencing data. Now there is a significant improvement even for the accuracy of long read sequences. For example, Nanopore, the time when we started and we sequenced Candida Swaja genome with Candida Swaja genome with the Nanopore sequencing methods. At that time, the base calling accuracy was somewhere near 70%. But then with the improvement in the base calling efficiency in the method, it's now probably close to 90%. So yeah, it's improving, but I would say the depth in the data and also an independent method of validation should be a good idea.
Yeah, and it can be tricky. I mean, aside from the fact that it's a diploid genome, so you've got two copies requiring increased depth, you can have strange ratios. So, yeah. I mean, ideally, you're going to have alleles of 1 to 1, but you can have 1 to 2 or 1 to 3, or
yeah, absolutely. And if you have the genomic regions that are they're present in multiple copies, for example, we found some that are repeatedly present, at least 3 copies or 4 copies. That even more complicates the matter, I guess.
Absolutely. Look through the group, check that whether there are any hands. What would be your hot tip for for people looking at antifungal drug resistance, and why there are these changes, and what induces Candida tropicalis to be more likely? What would be your hot thing to pursue next?
It's a very difficult question to answer, but I mean, in terms of course, there would be genome, but also their biology in terms of the reproduction, how they acquire the variants in the population, I mean, in terms of their genome organization. For example, Candida albicans has something in terms of their multiple repeat sequences, emergence, in their chromosomes. Candida tropicalis don't have them. At least from the sequence analysis point, I could not find the emergence in the Candida tropicalis genome. So there are differences, and it requires a lot of follow-up experiments to kind of validate whether that's responsible for the increased drug resistance that you see in Candida tropicalis. But and the other point that I want to mention is that, that when I started my PhD, at that time, people thought that Candida albicans is always associated with a patient, right? And then a few years later, somebody published the genome of Candida albicans from environment, right? And so we are learning new things every day, and I'm not sure what drives this difference, but I guess one has to look at different isolates and from different sources, different patients, different geography. It's difficult to answer what drives the difference, but you have to sequence a lot of species and kind of look through the genome probably. That's the way to do it, I guess.
More genomic work. Were you referring to the Oaks tree paper from Joe Dixon's group? Yeah. On Candida albicans. Yeah, that was a nice paper.
Yeah. Before that paper was published, I attended one conference, and somebody mentioned that they collected a isolate of Candida albicans from an apple, right? And nobody believed them. I mean, I don't know if it is true, but it's kind of interesting.
Mm. And the other thing I remember from that paper was the high number of SNP difference even between the same isolate when it was, highly.
Yeah, go ahead.
Oh, highly variable.
You mean within the same isolate?
Yeah.
Yeah. So I think there is some work going on in Gustav's lab after I left on the genomics part of Candida tropicalis. But I don't know if they found something or what's going on with that.
Any other questions? I can see everyone properly. Well, if there are no other questions currently, I think, Krishnendu, you might get some by email later, but thank you very, very much for coming and speaking to us all the way from Maryland. Really, really appreciate the time that you've set aside and your fantastic overview of this phenomenal work.
Thank you so much. I mean, it's really a pleasure to talk to you people, and it's always interesting to me to talk to new people. I did not know any of you before, except for you, Kathleen, but I'm glad to kind of meet all of you and know about the work that you are doing. Yeah.
Likewise, come to Australia sometime. Thanks again, Krishnendu.
Thank you very much.
Thank you. Great talk.
Thanks.
Thank you.
Thanks a lot.
Thank you.
Thanks.
Bye.
Bye-bye.